Good morning, doctor. Doctor, can you hear me?
Yeah, good morning, good morning.
Good morning. Students, we'll start our next session for today. Today we here with us is Dr. Gopalji Shah, Staff Scientist 5 from National Institute of Plant Genomic Research, Delhi. He was previously he was working as a scientist in CSIR Institute of Himalayan Bioresource Technology, Palampur, and also worked as a scientist in Sainsbury Laboratory, John Innes Center, Norwich, UK. He has numerous awards to his Swarna Jayanti Fellowship from DST, Government of India, Indian National Science Academy Medal for Young Scientists 2010, and NASI Award, National Academy of Science India Platinum Jubilee Young Scientist Award in 2007, and BIIS Fellowship from Department of Science and Technology, Government of India 2010-11, and a gold medal from Guru Nanak Dev University, Amritsar. He did his PhD in CSIR Institute, CCMB Hyderabad in 2018, and MSc in Biotechnology from Guru Nanak Dev University 2002, gold medalist. With this short introduction, I'll invite Dr. Gopalji Shah to deliver his talk. Doctor, the floor is yours.
Is my slides visible?
Yes, doctor.
And you can hear me also? Is full slides available?
Yes, doctor. It's okay.
Oh, good. Thank you so much. Thank you for the kind introduction and giving me the opportunity to talk about this important topic. So I was going through the course curriculum, and I could see that lots of undergraduates are the attendees. So I will be
mainly for undergraduate only. Doctor.
Yes. So though I'm going to talk about science and the scientific activities in my lab, but I'm trying to be very simple and try to emphasize you to how the microbes are helpful in sustainable agriculture. Okay. So in case if something is going fast and you are not able to understand it, please interrupt me at any time. Okay. So the title of my talk is Novel Approaches for Plant Disease Control and Growth Promotion. So we all know that we have been producing all the news channels and everything covers that we have been having enormous food production and that is enough. But remember that still around 800 million people go hungry every day. And we are also expected to produce 60% more food by 2050. So at the same time, around 1/3 of the total food produced per year, that is around 1.3 billion tons, is lost or wasted globally. And amongst them, around 20 to 40% of the loss is due to various diseases. So the emphasis is plant diseases are of utmost concern for sustainable agriculture, and they causes around 20 to 40% yield loss every year. And I just try to put some of the examples of the plant diseases. You might have seen rice dying with the disease. You might be seeing oranges getting wasted due to fungal disease. Similarly, there is a famous pathogen of apple, which is called apple scab. And also there is another fungal disease of rice called seed fly disease. So you don't need to remember the name of the disease. Just take it from me that there are many diverse kinds of diseases. They are caused by bacteria, they are caused by fungi, they are caused by insects, they are caused by viruses and nematodes and many other pathogens and pests. So controlling them is a very important theme for research activities and for agriculture. So thing is that we use lots of harmful agrochemicals to control the disease. I'm trying to show it here that a farmer is spraying fungicides, truck full of fungicides to control, for example, fungal disease in apple. And imagine a apple in the apple growing season, around 12 to 15 such spray occurs to protect the fungus apple from the fungal diseases. So I put a hypothetical image wherein I'm trying to emphasize that you can imagine an apple fully coated with fungicides. And these fungicides, though protect from the diseases, but at the same time is harmful for the environment because it can kill non-targeted organism, and also it is harmful for the consumers. So it's not a sustainable way for control of the disease. At the same time, we need to have the enhancement in the crop production. It's not like that we have to only protect from the diseases. At the same time, we should have continuous increase in some voice is coming. Please switch up your sound. So on the top of it, climate change, degrading soil health, emergence of newer diseases, pests, etc. are posing further challenge. So the take-home message is, though we have chemicals to control the disease, but they are not enough and they are not sustainable. So how to sustain agriculture without minimal use of agrochemicals is a challenge for us, our generation. So today I'm going to emphasize that how we can harness plant-associated microbes for sustainable agriculture. So our lab is working on one of the fungal disease of rice called seed blight disease. You can see the rice stem being necrotic, and I have zoomed this necrotic lesion here and trying to emphasize that once this pathogen infects rice, eventually the entire plant is dead and we got huge loss in the rice productivity. This disease is caused by a necrotropic fungal pathogen. For many of you, you may be aware of that there are different mode of colonization of plant pathogens. So one of the mode is necrotropic mode, wherein the pathogen will directly go into the plant, kill the plant cells, and eat on the dead tissues. So this is the mode of style of necrotropic pathogen. So our pathogen, Rhizoctonia solani, is a necrotropic pathogen, and it causes huge loss to the rice production. So around 70% losses occurs due to this disease, and there is no resistant genes known which we can deploy it for control of the disease. So disease management is a challenge for sustainable agriculture. So we have been trying to understand how to control this disease, and in the process, we could isolate a very novel bacteria from rice seedling. So there are many bacteria which live within plants. They are either largely associated with the plants, or they grow within the plant cells. So those which are growing within the plant cells are called endophytic bacteria. So this bacteria we found from the rice seedling being endophytic to the rice seedling. So this is naturally occurring, living within the rice plants. And when we isolated this is a yellow colored bacteria. And this is the Rhizoctonia solani, which I told you in the previous slide, that it is causing seed blight disease in rice. So interestingly, when this bacteria was allowed to grow together with Rhizoctonia solani, we saw zone of inhibition. So this bacteria is inhibiting this fungus to grow near it. So this is called antifungal activity, through which the bacterium is trying to prevent the growth of the fungi. Okay, so there are many antifungal bacteria known. But interestingly, this bacterium has unusual property to feed on fungi. In the next picture, I'm showing you this yellow color bacterium has now totally grown over the fungal mycelia and eventually eaten away the mycelia. So this is very interesting property wherein the tiny bacteria, so bacteria classically you may know they are very microscopic and they are prokaryotic. However, the fungus is multicellular and they are eukaryotic. So a tiny prokaryotic bacteria is able to eat thousands and bigger eukaryotic organisms. So this was a very interesting observation in our lab. So this property of bacteria wherein it can eat fungi is known as mycophagus. Myco means fungi, phagus means eating. So mycophagus property of the bacterium we could find out. So we take home message is that we isolated a bacteria which was living in the healthy rice plant, and this bacterium has an unusual property to eat this Rhizoctonia solani. So here I am trying to show you that during initial interaction, this bacterium, this thread-like structure is the microscopic bacterium. This is a microscopic image, and this bigger thread is the fungal mycelia. And so initially this bacteria has an ability to sense fungal mycelium, and eventually it leads to the degradation of the fungal mycelium. You can see it here. I am trying to show you that the bacterial treated mycelium is totally disintegrated. Okay, and further we used an MTT assay. This is a biochemical assay wherein we used a dye, MTT dye. So this dye will be metabolized by a living bacteria, living organism. So fungus Rhizoctonia solanae will metabolize MTT dye into a colored compound. So you can see formation of the colored compound in the living fungal mycelia. So Rhizoctonia solanae is living. That's why you can see appearance of this dark color. However, upon bacterial treatment, we don't see the Rhizoctonia solanae being able to metabolize MTT. So this shows that the bacterial treated fungi is dead. So I showed you two examples showing that the bacterial treatment disintegrates the fungal mycelia and also kills them. So this property of bacteria able to kill the fungal mycelia and able to utilize the fungal mycelia is called microphagy. So here I am trying to show you that if the Rhizoctonia solanae is allowed to grow in laboratory media, it will make a cottony mass. So it is a ball-like structure it forms. However, upon bacterial treatment, you can see this mass is drastically reduced. So this shows that this bacterium has not only killed the Rhizoctonia solanae, but has also metabolized it. It has used it as a source of nutrition. Further, we demonstrated that the growth of the bacterium is significantly enhanced in presence of the fungal mycelia. So here I am trying to show you that even in the laboratory media, this bacterium is able to grow to some extent. However, in presence of the fungal mycelia, the growth is significantly enhanced. So the way we estimate the bacterial growth is through colony counting. Maybe I think in your practical courses you might have by knowing it, that we need to do serial dilution of the bacteria and count how many bacterial colonies we get. So doing that, we observe that the growth of the bacterial cells are significantly enhanced in presence of the fungal biomass. So taken together, our results suggested that this NGJ1 bacterium, we need to isolate and identify the bacteria. So there are set of procedures which we follow to identify the bacteria. So I have not told you due to lack of time, but just trying to emphasize it. Generally, we do 16S ribosomal sequencing to identify the bacteria, but that will have sometimes difficulties in delineating the fungal genera. So we did even genome sequencing. So we did the genome sequencing of this bacterium, and using the genome sequence, we identified this bacterium as Burkholderia gladioli strain NGJ1. So this is a noble bacterial strain which is endophytic to rice and has an unusual property to feed on Rhizoctonia solanae. So I'm trying to emphasize now that this fungal eating property is not just specific to Rhizoctonia solanae, it is observed on many other fungal pathogens. So here it is a crowded slide. Don't try to look each of them, but just take from me that not only Rhizoctonia solanae, this bacterium can eat Fusarium, Alternaria, Magna porta, Venturi, and many others. Okay. So there are many types of fungal pathogens. So some will have chitin-rich cell wall, some will have cellulosic-rich cell wall. So those cellulosic-rich cell wall are known as Mycetes pathogen. So this bacterium not only kills the fungal pathogen, but it also kills the Mycetes cell wall. So this has a broad spectrum fungal eating property, and also the model laboratory yeast, Saccharomyces cerevisiae, and one of the opportunistic human pathogen that is called Candida. Even both of them were also killed by this bacterium. So this bacterium not only kills the plant pathogenic fungi, but also has the ability to kill the animal pathogenic fungi. So now, being scientists, our job is to understand, not only discover new things, try to understand and establish the scientific basis of this phenomenon. So I told you that we isolated and identified a very noble bacterium and a noble property of the bacterium. So there was nothing much known about this property. So our goal was to understand how a tiny bacteria can eat fungi. So for this, we try to understand the mechanistic detail, and for this we do RNA-seq analysis. So for the benefit of the students, I don't want to go into the detail how we do RNA-seq analysis, Maybe if anyone of you are interested, we can discuss at the end of my talk. But just to tell you, this is a very, very strong tools through which we can identify what all are the differentially expressed genes, what all are genes which are particularly upregulated during that particular interaction, and which was downregulated during that particular interaction. So using RNA-seq analysis, we did bacterial fungal transcriptome analysis and try to identify which genes are differentially regulated. So interestingly, we observed a set of genes where, again, you don't need to remember what are those genes, but you just take it from me that these set of genes are required for develop synthesis of a secretion system. So that is called type 3 secretion system. So bacteria is a gram-negative bacteria, and if you see the cell wall structure of the bacterium, it has periplasm, it has cell membrane, and also now it has to interact with the host. So imagine this is fungus, this is bacterium. So this bacterium is trying to make a needle-like apparatus, which is called type 3 secretion system, through which it can send protein directly, inject protein directly into the eukaryotic host. So what we observed that this particular secretion system is upregulated during mycophagous interaction of NGJ1 with arsenite. So this was very interesting, as the role of this type 3 secretion system was not being reported in any bacterial fungal interaction, but it has been found to be an important virulence factor for animal pathogen and plant pathogen. So people have shown before that a plant pathogenic or animal pathogenic bacteria uses this T3SS needle-like apparatus to deliver effector proteins directly into the eukaryotic host and kill it. But our data shows that this particular secretion system is also upregulated during mycophagous interaction. So it was very interesting, and we tried to study its function. So how to study the function of a gene? We need to mutate we can do either loss of function and gain of function. So ideally, you can try to mutate the gene in your organism. Now the bacterium will not be able to make that gene. And if that is deficient in that process, you can correlate, yes, indeed, this gene is essential for this phenomenon. So using this simple hypothesis, we try to mutate one of the genes involved in type 3 secretion system formation. And when we disrupted this gene, now the bacterium was Microphagy deficient. You can see this is the wild type bacterial treated fungi, and you can see bacterium has totally eaten away the fungi. However, this mutant treated fungi is not killed, and you can see the fungus being grown on the plates and producing spores. So this is a clear demonstration that type 3 secretion system is important for mycophagy. And I told you, as it can, previously people have shown that type 3 secretion system is required for bacterial plant interaction. So when we disrupt, we infiltrated the wild type bacteria in tobacco leaves, we see a hypersensitive response. This is a hallmark of plant defense response. So people have shown before that if you take any non-host bacteria into Benthamiana, it induces the defense response leading to cell death. However, this T3SS mutant bacterium was unable to induce the cell death response. So this clearly demonstrated that indeed our mutant was type 3 defective, and also it was effective in mycophagy. So this is a very interesting phenomenon which we detected. So now how this type 3 secretion system is helpful for mycophagy? So that was our next question. So I told you there are many proteins which bacteria will take it and deliver into eukaryotic host. So we try to identify what all are those proteins, bacterial, they are called effector proteins. So what are those bacterial effector protein which is deployed to kill the fungal cells? That was our next question. So for this, we used the genome sequence and computationally predicted what can be these effector proteins of our bacterium. And we identified around 35 such effector protein which is encoded in our bacterium. So now to demonstrate whether they are indeed secreted into the fungal cell, we try to investigate detail. So here I want to emphasize it that amongst those 35 effector protein, we identified one particular protein which has homology to a prophase tail-like protein. So this protein amongst one of the prophase tail-like protein was predicted to be the effector. So here I am using a scientific term, prophase. So I'm sure many of you may know that in bacterial genome, there are bacteriophages which are in the lysogenic or lytic cycle. So the bacteriophage genome is integrated into a bacterial genome and they are there. But under certain conditions, they will be expressed and the bacteriophages will be formed and that will kill the bacteria. However, in this case, we identified a prophage like protein. Actually, that is a bacteriophage protein, and that was predicted to be a T3SS effector. So it was very exciting that how a bacteriophage protein can be a effector to kill the other bacteria. So naturally, it was made to kill the bacteria, but now it seems that the bacterium has learned to use it to kill the other fungi. So that was our excitement here. So to investigate further, we first made an antibody specific to this protein. And so we have antibody and we demonstrated that indeed our bacterium delivers this prophage like protein into fungal mycelia using this secretion system. So this is the Western blot image, and don't for the graduate student, it's not very essential to understand the theory of it, but just take it from me that this confirms that the wild type bacterium delivers this BG9562 into the fungal cells and kill it. However, if the type 3 mutant bacterium is not able to deliver it. So with this, we prove that 9562 is a bona fide T3SS effector of NGJ1. So now next question was whether this 9562 is important for microphagy. For again, we use the bacterial genetics approach wherein we mutated this 9562 gene in this bacterium. I told you that the wild type NGJ1 bacterium kills the fungi. It is microphagy. But when we mutated this 9562, this mutant bacterium is unable to kill the fungus and is unable to grow over the fungal mycelia. So this clearly shows that 9562 is important for microphagy. So further we express the full length copy, the wild type copy of the gene in this mutant bacterium, and now it restores microphagy. So this is a clear demonstration. This is a clear demonstration that 9562 is important for bacterial microphagy. So next question is that how this 9562 is killing fungi. So that is important question. So we thought that maybe it may have some toxic domain or any enzymatic or lytic domain which may be degrading the fungal cell wall and killing the fungi. So for that, we used a computational approach and we did a phylogenetic analysis. So ideally, this 9562 protein showed homology with the prophage-like protein, but none of them has any toxic and enzymatic activity. All of them has a phage activity. So this shows that though it is related to phage, but it does not have any lytic activity. So question was how it is killing fungi. So for this we observed that this gene is encoded in a bacteriophage cluster. So there are bacteria. I told you that bacteriophages are inserted in the bacterial genome. So there are set of genes which is required for its lysogeny, DNA replication. Some are required for head assembly, some are required for tail assembly, and some are required for lysis of the bacterial cells. So interestingly, in our bacterium, we observed that lysogeny, tail assembly, and lysis genes are encoded, but head assembly genes are missing. So this is very interesting. So this shows that bacteria has deleted the head of the bacteriophage and utilized one of the cryptic bacteriophage gene for killing the fungi. So this is what we thought. And to further to rule out the involvement of bacteriophage, we try to see whether any bacteriophages are induced during microphagy. We could not see. So this clearly hints that there is no involvement of bacteriophage, only this protein is deployed to kill the fungi. So interestingly, we observed that there is a particular around 9 amino acid sequence which is only present in our bacterial protein sequence, but none of the related bacterial protein sequence has this secretion signal. And I told you this was a secreted protein, so we felt that these 9 amino acids at the end terminus of the protein is important for secretion and delivery into the fungal cell. So we clone the full engine sequence and we also clone the deleted variant wherein the secretion signal is deleted. So when now we express this plasmid into the bacterial cells, so I told you wild type bacterium is microphagy, the mutant bacterium is not able to feed on fungi. However, when we express the full length copy of this plasmid, PGD3 plasmid, wherein the full length gene is expressed, the bacterium is again the microphagy. But when we express the signal deletion variant, the bacterium was not getting microphagy. So this is again a clear demonstration that the type 3 dependent secretion of this BIIS9562 is important for microphagy. So to further prove it, we took this gene and overexpressed in another bacterium. So that is Ralstonia solanacearum, so which is otherwise not microphagy. So this is the wild type bacterium treated with fungi. The Ralstonia is not growing over fungal mycelia. But when we express this 9562 in Ralstonia solanacearum, we started seeing the bacteria growing over the fungal mycelia. And when we disrupted the type 3 secretion system of Ralstonia, the type 3 signal mutant bacterium is not able to forage over the fungi. So this again confirms our hypothesis that indeed 9562 is important for fungal eating property of this bacterium. So to prove this further, we purify this protein. So again, we are using a new tool it's a routinely used tool to purify the protein. There are some bacterial expression system in which you have to clone your gene of interest. It will transcribe and give you the protein, and you have set up things to affinity purify the protein. So we purify the protein and see whether the purified protein can kill the fungi. So here I am trying to show you that the buffer treated Rhizoctonia solanae is growing well in the laboratory plate. So this is quite usual. But when we saw the 9562 treated fungal cells, it was not able to kill the fungi. It was killing the fungi directly. You hardly see any fungal growth on the protein treated cells. And similarly, we observed that the protein treatment leads to the disintegration of the fungal mycelia. You can see the fungal mycelia being disintegrated upon protein treatment, while upon buffer treatment, the mycelia is quite intact. So this shows that the protein 9562 purified protein can be antifungal. And on Rhizoctonia solanae, we further tested whether this antifungal activity is common to other fungal pathogens as well, and we observed yes, the antifungal activity associated with 9562 is universal. It's a broad spectrum antifungal activity. At the same time, this purified protein does not have antibacterial activity, so it did not kill any of the tested bacterium cells. So this shows that this purified protein can be used to control fungal diseases of plants and animals. So what I'm trying to emphasize is how a fundamental biological questions, how a fundamental biology can give you to help you to innovate a newer solution to the problem. So with all this basic research, we identified a novel antifungal protein which can be used to kill the fungal pathogen of plants and animals. So first I will try to summarize the biological observation which we had till now. So what I told you that there is bacteria which in the plant there are lots of bacteria and fungi living together. So this NGJ1 bacterium has an ability to sense fungi, and upon sensing the fungi, it uses type 3 secretion system. I am trying to put this needle as a type 3 secretion system through which it will deliver effector protein into the fungal cell, and one of them I told you we identified to be 9562. So this 9562, once it is delivered into the fungal cell, will lead to the lysis of the fungal membrane and releases fungal metabolites into extracellular milieu. And now this fungal metabolite serves as a nutrient source for the bacterium. So now the bacterium can grow over fungi. So this is helpful for the bacterium not to damage the plant, to obtain the nutrient and utilize endophytic fungi to use it as a nutrient source. So it can be a very good property of the bacterium to avoid harming plants, at the same time using the harmful fungal pathogens and use them as a source of nutrition. So that means it will help the plant to protect against fungi, at the same time will not be able to harm the bacteria. So this my plant. So this property is called microphagous interaction, and we are trying to use whether microphagy can be used to control the plant diseases. So this work was published in a prestigious journal called Nature Communication. So basic part of it, we published this. So now I told you that this 9562 is an novel antifungal protein. So now we have started trying to utilize, see whether this purified protein can be used to control the seed fly disease in rice. So for that, we have obtained a variant of this 9562, which is thermostable, more stable, and more active. So this is patent protected in USA, China, Indonesia, and patent application is under examination in India. So this patent protected variant, we are now trying to use it for control of the disease. So here I am trying to show you that if you take this protein solution and spray onto the rice plants, if there is no protein and the Rhizoctonia solani is causing disease lesion, so the buffer treated one, you see the characteristic disease lesion of Rhizoctonia solani. However, upon protein treatment, you see very reduction, almost 95 reduction of the disease severity, and we only see marginally a smaller necrotic lesions, and that will not spread further. However, in the buffer treated ones, the disease has even spread up to the leaves, and eventually the entire plant is dead. To show this, indeed it is happening, we saw that the protein treated mycelia is dead, while the buffer treated mycelia is able to happily colonize the plant tissues. So this shows that the protein can be effective to control the fungal diseases. So this was under the in vitro condition, in the greenhouse condition. Now we started trying it in collaboration with IIRR Hyderabad. This is the ICR Institute in Hyderabad, and IARI, again ICR, Premium ICR Institute in New Delhi. So their pathologist help, we are trying to develop an antifungal formulation using this protein to be used as a spray to control the seed fly disease. So under field condition, you can see water around the field. So under field condition, this is the control Rhizoctonia solani. You can see the disease there, but the protein treated ones, there is no disease. So this suggests that the protein treatment is going to be helpful for control of the disease. But there are still challenges because mostly the rice grows in the rainy season and there are lots of water. So the spray will be lost. Most majority of the spray will be lost in water. So we need to have the effective delivery systems, and for that we are trying to develop nano formulations and emulsification so that the protein can be effectively deployed to control the diseases. Another means to deploy the protein is using transgenic. So here what we do, we try to overexpress this bacterial gene into the plant and see whether the plant is resistant to these diseases or not. So we made a transgenic rice plant where this 9562 was overexpressed under pathogen-inducible promoter. So this is an empty vector plant. You can see the upon pathogen infection, the entire plant is dead. However, both the transgenic plant, two independent transgenic, we have another transgenic events also. I am showing you only two important events, and in both of them, there is practically zero disease. So this shows that mere overexpression of a bacterial 9562 protein provides seed blight disease tolerance in rice. And now we have done transgenics in other crops and we observed that the overexpression of this provides multiple fungal disease resistance in rice as well as other crops. So this is an effective solution for the disease control. So I told you this 9562 has a strong antifungal activity and can be used to control the fungal disease. So a bacterium, a plant-associated bacterium is helpful for the control of the disease. So I hope until this is clear to you, and if you want to have 2 minutes questions, we can take it now, or we can discuss at the end of it also, and I can take something else now. So is it okay? Shall I proceed or shall I wait and discuss?
Students can respond if you want doctor to continue to the next topic.
Okay, maybe at the end we can discuss.
At the end we can do, doctor.
Yes. So till now I told you how an antifungal property of this bacterium is helpful for control of the fungal disease. So recently we observed that beside antifungal property, this bacterium, this NGJ1 bacterium, also has a strong antibacterial property. So earlier I told you that it has ability to kill many fungal pathogens. Now I'm showing you that it has ability to kill other bacterial pathogens also. So for example, this NGJ1 bacterium is antibacterial to E. coli, Agrobacterium, and Xanthomonas. And here you can see this zone of inhibition around this the yellow colored bacterial colonies. So this signifies the antibacterial activity. So antibacterial activity is observed on E. coli, Agrobacterium, Xanthomonas, and many other rice-associated endophytic bacteria. So this is very interesting that one single bacterium can kill fungi, can kill other bacteria which lives together in the rice endophytes. So again, we try to understand what is the basis of this antibacterial activity. And due to lack of time, I am not going to go into the detail of antibacterial activity, but just I want to summarize the finding using this cartoon. What we observed that bacterium has two different weapons to delivers various toxin antibacterial proteins into the prey bacterial cells and kill them. So for example, this NGJ1 bacterium is having one weapon called T6SS1, another weapon called T6SS2, and through which it uses many very effector proteins to target, like for example, this bacterium, this bacterium, this bacterium, this bacterium. But there are some bacteria which has immunity, which have shield against this effector protein. So some of the bacterium it is not killing. So for many rice-associated bacterium, we observe that the NGJ1 is killing them using type 6 secretion system 1, T6SS1 and T6SS2 dependent manner. And we identified a novel evolution in bacteria wherein bacterium has converted its own toxin. Actually, this toxin was made to control bacterial own growth under certain environmental conditions. But now this bacterium has learned to utilize this toxin protein to kill other bacteria. So this work is again published, and I have shared the PDF file with the organizers. So hopefully they may provide to you. You please go through it. And in case you have any doubts, you can even write back to me and send to the organizer, and I'll be very happy to clarify your queries. So the take-home message was that one single bacteria which was isolated from rice can be so helpful to not only protect the fungal diseases, but also the plant bacterial diseases as well. So this was the message. But now I'm trying to emphasize that there are hundreds and thousands of bacteria, millions of bacteria, which are living within and in association with plants. So large numbers of microbes live within and in association with plants. So one microbes it can have such a profound impact on the plant health. So imagine how these microbes might be interacting together and interacting with the plants to promote their growth and surveillance. So that is interesting. And recent work is coming up which suggests that indeed these microbes have profound impact on plant health, growth, and development. So now the question is, can we harness this microbiome for sustainable agriculture? And the answer is yes, we can harness these microbes for sustainable agriculture. To again re-emphasize these points, I'm trying to show it here that these microbes are in soil, so they are associated with the roots. They are called rhizosporidic microbes, and the microbes which is inside the plants are in the phyllosphere. They are also in association with the plants, and so these airborne microbes, soil-borne microbes have some affinity with the plants. They live in association with the plants, and I'm trying to propose, and the people are trying to prove that indeed these microbes are helpful for the plant growth and development. The question is how to harness them. So people have shown that I've shown you examples of this Burkholderia and how they are antifungal and antibacterial. So similarly, many other researchers have shown that indeed the plant-associated bacterium are helpful. For example, this Bacillus, which is again a plant-associated bacterium, can be antifungal, and people have found the mechanism of antifungal activity. Again, this is antibacterial. It can regulate the motility and bioformation ability of other cohabiting bacteria, and also it acts as an elicitor of defense response. ISR response is a kind of defense response in plants. So a single Bacillus strain can not only kill the fungi or bacteria, can also induce plant defense. So ultimately, it can immunize plants against subsequent bacterial infection, bacterial or fungal infection. That's the message. So people are trying to use microbes as a biopesticides, as biofungicides, as well as biofertilizers. So again, I have shown you, shared one recent review article from my lab. You can read it to see how microbiome functions are helpful for the agriculture. But now the question is that how to harness microbiome. So here I am telling you one example. So one bacillus, one Burkholderia is doing it. But there, I told you there are millions of such microbes, so many of them will be beneficial. So if you can isolate those beneficial microbes from the plant and develop a synthetic community of the microbes, that is called syncom. So if you can design syncom of beneficial microbes, it can be very helpful for sustainable agriculture. So people have shown that use of syncom is promoting tomato growth. For example, this is a control tomato growing to some extent. However, upon this synthetic community treated treatment, this tomato is growing extensively. So this clearly shows that under field condition as well as under the lab conditions, the synchromes are working well and it's helpful for sustainable agriculture. But there are challenges also, that how to design the syncom, because imagine if you are in south, you may have different agroclimatic regions. If you are in Ahmedabad, or if you are in Delhi, or if you are in Patna, the climatic regions are different. So the microbial efficiency to colonize that region of soil or plant may be quite different. So there are extensive research is needed to understand how microbes colonize plants, how microbes colonize soil, what all are the environmental factors which modulates, and what kind of microbes can be universally grown across the agroclimatic regions or across plant genotypes. So this is the need of the time to understand how these synthetic microbes can be used to sustain agriculture. Another challenge is that many of the bacterium or microbes may not be cultured. So you might be aware of that only a handful of microbes we can culture, and the majority of them we are not still able to culture them. So the question is how, if we can even develop tools to culture them, it will be very helpful. So thanks to genomic approaches, we can have the microbial genome sequence, consortium genome sequence, and try to understand why these microbes was not being cultured. So if we can identify those unculturable microbes and develop culture practices for them and use them for synthetic community, it is going to be helpful. So microbiology, computational biology, artificial intelligence driven synthetic community altogether is going to have a huge impact on the plant growth and sustainable agriculture. Another important development is for phenomics. So ideally, right now we have limited tools to study one plant or set of plants. But imagine we have huge lands, huge fields. So how to keep monitoring them individually? So if we have some phenomics tools which can be monitoring our crops without our eyes, it is going to be a future innovation. So in future, we are going to have a huge demand of microbial genetics, genomics, artificial intelligence, development of synthetic communities, and phenotypic facilities to use synthetic community for climate resilient crops. So with this, I'm coming to the end of my talk. So before that, I should acknowledge the fundings from different various funding sources. I've been generously funded by Department of Biotechnology, Department of Science and Technology, NIPGR, and also Swan Jayanti Fellowship from Department of Science and Technology. And I acknowledge the hard work of my students. So the microphagy work has been done by Durga Madhav Swain, Sunil Yadav, Rahul Kumar, and many others. And similarly, the antibacterial work has been predominantly done by Sunil Yadav. And we all work together in a team and try to understand how we can use microbes for sustainable agriculture. Thank you. So with this, I will be happy to take questions.
Thank you, Dr. Gopal. Thank you for your presentation today. So students, if you have any questions or clarifications needed on the presentation, you can ask Dr.
Sir, somehow voice is breaking actually, not able to hear it.
The connection is unstable, I think.
Yeah, yeah.
Any
Students,
queries,
please.
students? You can also use the chat box to ask questions.
Yes, yes. So can you hear it? I can't hear it. Students, please put the chat box or talk. Switch on your mic, camera and talk.
Good afternoon, sir.
Yeah.
I have a question. Is there any possibility of the fungus getting, like getting resistant to the bacterium?
Yes. This is
So-
possible. Yeah, very good question. This is possible. Generally, they are living organisms, so they may be evolving resistance against even bacteria. So though it will be hard because the bacteria may have multiple approaches to kill fungi. So even resistance against some of the means may not be enough for development of resistance. But indeed, it is possible. And people have shown in other antifungal bacteria. So there is another genus called Colimonas. So this Colimonas is also microphagous. It can eat fungi, and people have shown it that if you can mutate this fungal pathogen, which fungus which is being killed by Colimonas, then you can expose it to radiations and then identify fungal mutants which is now resistant against Colimonas. So they are trying to understand how now they have this resistance has become, but indeed it is possible. And similar is true for, let's say, disease resistance or antimicrobials, like we spray fungicides to kill the pathogen, but pathogens evolve fungicide resistance. So that's why biology is challenging, and we need to have a ever-growing efforts to understand the biology and contribute. One solution is not long-lasting. We need to have backups.
Yeah. Thank you, sir.
Thank you.
Any other questions?
Sir, I have one more question.
Yes.
Like again, it's a bit technical. So you said like you will be able to explain the RNA sequence analysis for like detecting like the T3SS secretion system. So can you elaborate a bit about the analysis?
Sure. So do you know how RNA-seq works, or no, you don't have idea right now?
No, I don't have an idea right now.
Okay, good. So no problem. So I will be very simple and try to give you in a layman language. So ideally, you know that any cells will have first transcription. It will make RNA, and that RNA is produced into the protein, and then protein do functions. Okay. So to understand what all are the RNAs which are expressed under the given condition has been always ever long questions for the biologists. So question is to identify those RNAs which are expressed under given condition. Okay, so now condition can be growth, condition can be upon, let's say, this bacterial treatment, condition can be a cancer patient, condition can be a COVID patient and things like that. So we need to identify what all are the transcript genes which are induced, are expressed, are downregulated under that condition. So you should have control. That means those organisms which is not subject to that, and another one of treatment. So control and treatment sample you take, isolate total RNA. So you can isolate total RNA, and in that you will have mRNA pool. So total RNA will have the mRNA pool. So now those mRNA pools, ideally, traditionally people used to do microarray. So wherein they have the chips, wherein you can see which all are the genes which are expressed. But now with technology, we can sequence without knowing what RNA it is. We can entirely sequence the mRNA profile of that organism. Okay, so there are many tools now. So they are called next generation sequencing approach. So traditionally people use Illumina sequencing to identify shorter reads of each of the RNAs. So you know these sequences are expressed. Now computationally you can assemble them together and see this is the transcript which is assembled. So you will have control transcript, you will have the treated transcript, and you can computationally now identify that these transcripts are more expressed in this condition, these transcripts are more expressed in this condition, and this transcript is underexpressed under this condition. So this is in nutshell RNA-seq analysis. So we use next generation sequencing approach to sequence the entire mRNA pool and then use computational biology approach to identify differentially expressed genes. So using this tool, we identified that the type 3 secretion system was expressed, upregulated during microphagous interaction. So this is in nutshell RNA-seq analysis.
Thank you, sir.
There's a question in the chat box.
I can't see the chat box. Okay. What is northern blotting? Is it different from RNA sequencing? Yes, very good question. So you nicely figured it out that if the goal is just to see the RNA transcript, we can do blotting. So northern blotting. But that northern blotting is one gene at a time. So if you know the gene, then only you can blot for that gene. Am I right? So northern blotting, what we do, that we isolate mRNA, blot it, and then look for whether that particular probe is expressed or not expressed. So northern blotting is used to show that particular gene is expressed or not expressed. But idea is we don't know what genes are expressed. So we are blind right now. So in the ocean, we are trying to identify what genes are expressed. So for that, we do RNA sequencing. Here we are not only targeting to study one gene. So we, without knowing what genes they are, we amplify all the mRNA, we sequence all of them and try to then predict what mRNA has been expressed or downregulated. So that's why, though they are similar to techniques to know whether transcripts are expressed or not expressed, but northern blotting is a very traditional time-tested approach to show a particular gene or transcript is expressed. But RNA sequencing, without knowing what is what, we sequence them and then predict that yes, these sets of these 100 genes are expressed under one condition, these 200 genes are expressed under another condition, and this is how they're dynamically evolving. So that's what. So they are related but not same. Yes. So another question is, please explain about plant microbiome and mechanism behind it, and also what kind of microbes used. So I told you what is plant microbiome. Microbiome is set of microbes which are associated with the plants. So now these microbes can be living within plants or can be attached, growing on the plant surfaces. So they can be epiphytic, they can be endophytic, they can be root associated, they can be phylosporic associated, shoot associated. So the entire sets of microbes which live within or in association with plant is called microbiome. So this is plant microbiome. So now imagine by birth all the microbiomes may not be there. So this is a very interesting feature of the microbiome. So like genetics, we acquire all the genes from our parents. So this is genetics, but we acquire microbiome as through our lifespan. So the plant acquire microbiome through growth during the environment, and this microbiome has, imagined that many millions of genomes are there now. So the coding potential of the microbiome will be more vaster than the coding genetics which we have inherited from our parents. So the concept is emerging that microbiome is going to have a profound impact on the health and development of any given organism, no matter plants, animals, and things like that are human. Microbiome is coming up as a bigger guardian in this picture. So now question is how microbiome is shaped, because by birth we have not inherited from the parents. So what it seems that during growth, the food we eat are in plants, the soil where they grow or the atmosphere they grow, they acquire this microbiome. Okay, so it seems that plants releases some root exudates. These root exudates attract the beneficial microbes towards plants. Now there can be pathogenic microbes also. So these microbes compete with each other, try to prevent the beneficial, the pathogenic ones. Micro beneficial ones will colonize and can be contributing to the healthy lifespan of the plants. But under conditions are during climate change, this imbalance of the microbial consortium is getting disturbed, which causes dysbiosis. There is imbalance in the microbial community, and this dysbiosis leads to abundance of the pathogenic organism that causes disease, and ultimately we lose this plant. So besides plant genetics, this symbiosis or dysbiosis of this microbiome is a major player which contributes to the plant disease and growth development defects. Thank you. And which kinds of microbes are used? Now, there can be any kinds of microbes. There can be bacteria, there can be fungi. So I told you a few examples. Our own study shown that Bucholleria is helpful. I talked about a previous study where bacillus had been extensively used. So these are all bacteria. Then there are fungi. So there is the famous trichoderma. So this is a plant beneficial fungi which is doing similar job as done by the bacillus or Bucholleria to promote plant growth. Similarly, there is another fungus, Pyrnospora. So they are known to promote the plant growth. And now extensive research is going on to identify more and more beneficial bacteria and fungi which can be contributing to the plant growth and development. So huge source of microbes there.
Any more questions? So I guess no more questions. Thank you, Professor Dr. Gopalji, for your presentations. It is very good to know a lot of things from the interactions between plant and microbes. And Doctor, you can also share your presentations with us. We will pass it to the students along with your papers, research papers you shared today morning.
Sure, I will
if any
do that.
questions is there, I will get back to you through your email.
Sure. Thank you so much. I will do that. Thank you.
Thank you so much, Doctor.
Okay, thank you. Good day. Bye. Bye, students. Thank you for listening. And I hope you are motivated to look microbes as a different tools and exciting tools of research and contribute to the sustainable agriculture. Thank you so much.
Thank you.
I'm logging up now. Thank you.
Sir, can you drop your mail here?
Students, that's all for today. We are winding up. I will send you the links for the papers presentations that doctor has shared with me. You can download them and read them. And if you have any questions, you can communicate with us through email or WhatsApp. Okay, we will get your answers from the concerned persons and we will give it to you. Okay. Thank you so much.